of the GFRA. And one of the goals of the GFRA is to provide periodically some information, updated information on the status of research. And that's what this workshop was about. The idea was to gather together specialists and different people working on the EuFMD problem to try to identify which were the main gaps on research on food and mouth disease. So this is the picture. This meeting was done in Argentina during June this year. Actually, this was not the first one done in Argentina. There was a previous one that was accomplished in 2010. As you can see, younger people and a fewer number of people also. And actually, Cyril Gay told me that there was a previous one in 2006. We don't have pictures of that one. But if we follow the trend of fewer people and younger people, it could be something like that. But I mean, we don't have really strong evidence about this. So the idea here is that this is a list of attendants. As you can see, there were researchers from different areas of the research, of knowledge, but also officials from government institutions, regulatory officers, both from Argentina, but also international, like OIE, Panaftosa, and also a strong presence of vaccine manufacturers, people that work in companies that make vaccines. As you can see, there were 4 of them. And the idea also was to have a geographical representation that could cover more or less both endemic settings and countries that are free or free under programs, like, for example, Argentina or Brazil. So let's go just to give you a general idea of how was the meeting. It was 2.5 days, very intensive 2.5 days, let's say. And we prepared different blocks, as you can see, in different topics, as virology, diagnostics, host pathogen interactions, both from the host side and also from the side of the immune response also. Also a block on vaccines, different aspects of the vaccines, and also in the last day, epidemiology. And as you can see, each one of these blocks, they have slightly different issues. And there were 2 coordinators that will give an introduction of state of the art of this topic, but this time, and later on coordinate the discussion, because the main goal of this is to have a room all together to discuss about these topics, and not to give like keynotes presentations, but you know, to get an open discussion on the issues. So according to the topic of this open session, we will just focus on 2 of main sections, that was vaccines and immunity. So what are the current obstacles? Actually, many of them, they have been with us for quite a long time. And as you can see, well, I can go really fast on them, but we know that conventional vaccines cannot be produced outside BSL-3 containment facilities, and they have a narrow serotype specificity. I mean, the range of protection is really restricted to certain strains, plus the antigenic drift of a virus many times require to update whatever it's stockpiled in the vaccine banks. Also, that the current vaccines do not provide immediately immune protection, but it allows a window of between 4 and 7 days where the animals are still vulnerable to the infection, and also they do not provide a long-lasting immunity. Also, the vaccines, or another shortage, is that they do not provide a sterile immunity. That means it does not prevent subsequent infections forever, and it does not prevent the establishment of persistently infected animals. Another problem is that the ecological settings and the regional needs probably are different, and mainly the vaccines are more or less the same in terms of strategy below the formulation. For some vaccines, the presence of non-structural proteins really it's a problem for the implementation of strategies based on differentiation between infected and vaccinated animals. And these last two are closely related, but both of them, they talk about the absence of standardized vaccine quality controls that are applied to all the vaccines all over the world. But that does not happen. I mean, it depends on the country, it depends on the region, it depends on the producer. The test, the potency test, purity, strain identity, antigen payload, may be done, maybe not. It may be informed to the buyer, maybe not. So that's also a problem that should be somehow tackled. So what have we done, or what have the science done during these years to tackle some of these problems? First, the conventional vaccines, let's name it like that, they have been improved in terms of the methods to concentrate the antigen and to purify it, so that allows that many of good quality vaccines, they are high potency DIVA vaccines. They can be used like that. Even they are not engineered to do that, but as long as they really get rid of non-structural proteins or during the process, it can be used as DIVA vaccines. Then I will show you three blocks of three other approaches that has been around for some times. And tomorrow we will have a whole session on all these three experimental approaches. One is the adenovirus vector vaccines that has been tested in cattle and swine with good protective results. And also from the first idea that was around a serotype, now we have different constructions for A, O, Asia1, and SAT2 serotypes. Also, they have completed minimum dose immunizing doses, and a vaccine release dose has been approved in the United States. Plus, this strategy allows the combination of other adenovirus vector proteins, in this case interferons, that may promote a protective state during the first day when the window of vulnerability is still on for the conventional vaccine or for the adenovirus vector vaccine. Another platform are the VLP-based vaccines that they have also went through a long road, but at this time we can say that there are reliable platforms to produce them, both in baculovirus and in mammalian cell cultures. The new genes constructions to produce VLP-based vaccines allow to easily swap between for producing VLP from different strains, and also they have improved the production yields of these VLPs. The structural studies on the viral capsid also allow to reveal new strategies to improve stability for selected strains. And they have already been tested in target species with good results. Also, another approach that has been also developed in Plum Island, it's the DIVA marker Cdna derived inactivated FMD vaccines that include negative markers for DIVA use. It's a leaderless, so it's a highly attenuated virus, although it is used as a killed antigen. It's also designed for swapping between sequences. And the backbone is derived from a strain that grows really well in BHK. So that could be adapted to the current manufacturing processes. So what are the gaps? Testing and development of new adjuvants and formulations. So that would improve the interaction between private sectors and researchers. Vaccine promoting necessary immunity, new vaccine delivery methods, and define immunological relevant parameters that will allow to get an indirect assessment of vaccine efficacy, avoiding challenge of animals in different target species, and also the design of fit-for-purpose vaccines according to the animal species on ecological, on geographical settings. In the immunology side, we have point out a number of advances since 2010. Many of them, they are regarding the response in the target species in cattle and a little bit less in swine. But this also gives a new perspective of what is going on when you vaccinate or when you infect animals. What are the immunological processes that are put on stake in that situation? What are the gaps? Well, they are related to the ones with the vaccine. First is define really what are the immunological mechanisms besides the antibody production that we know that is the tool, but we have to know how to produce this tool more efficiently, both in protection against homologous and heterologous infections, and study the mechanisms that lead to the establishment of memory responses, and that these memory responses can remain a long time. Also define the importance of cell-mediated immune response and the innate immunity, both as to generate an antiviral state, but also as a link with the adaptive response, because this is also a key aspect. Identify immunological parameters induced by multivalent vaccines, vaccination schedules, antigen payloads. And these last two are really things that are starting, but they involve big data analysis for identification of genes that are involved in the immune response. There have been some recent works on this, and I will comment a little bit on them tomorrow. But that will help to understand what makes a response protective and help design new strategies to go towards that kind of response. And also all the structural studies on the interactions between antibodies and antigen that will provide molecular basis for vaccine design. So this is basically it. This report will be soon available at the GFRA site. And this is just the acknowledgement to all the participants, the USDA and the INTA, who had support financially this meeting. And of course, well, I'm open to any questions, and thank you very much for your attention.